Welcome back for more physiology. We're going to be dealing with kidney function today. And in particular, we're going to look at filtration pressure and active transport in terms of how do we filter your extracellular fluid. So the first thing that we should probably look at are the pieces of anatomy in terms of what goes on with urine formation. So we all obviously have two kidneys. The one on your right side is going to be lower than the one on your left, and that's because you have a liver in the way. We're going to assume that you have two kidneys, even though you could have more, and it won't do anything to you, just happen to have more kidneys. Each of the kidneys will be connected by a tube called the ureter to the urinary bladder. The urinary bladder is just a big ball of smooth muscle, which means as it stretches, it will contract back down, and then it'll be attached by two sphincters, an internal and external sphincter, to a tube that we call the urethra, which is where urine then flows on out. Seems simple enough. So if we look inside of a kidney and you've dissected them, if I were to look inside of the cortex and the top of the medulla, and in particular within a pyramid, what I would notice if I zoom in are these tubes. And the construction of how we figured this out is all fascinating and all that stuff. But what we notice is tubes all over the place. So what we'll end up having is we'll have an arcuate artery, we'll have an interlobular artery, and coming off of that we'll have an afferent arteriole, which is then going to feed into a bunch of capillaries that we call the glomerulus, which will then have another arteriole leaving it called the efferent arteriole. So we have a capillary bed with arterioles on both sides. That will be connected to a big old network of capillaries that we call either the vasorectal or the peritubular capillaries, depending on the location. Those will then feed into a venule that will then attach itself to an interlobular vein, which will attach itself to an arcuate vein, which will then move its way on out. If I look at the tubes itself, the glomerulus is trapped inside of a space called Bowman's capsule or the nephrotic capsule. So that is technically the start of all the parts for the kidney, or this functional unit that we call a nephron. So fluid captured by the Bowman's capsule will go into a series of tubes that are all over the place that are called the proximal convoluted tubules. Proximal because it's the first set near the start, convoluted because they're going all over the place, and tubule because it's a tiny tube. Attached to that, we're going to go into something called the loop of Henle or the nephrotic loop. It's not as super functional with us, but in other animals it's very functional. As we head back up towards those peritubular capillaries, we're going to have another set of tubes going all over the place, another set of convoluted tubules, but this is near the end, so we call that distal, or the distal convoluted tubules. That will terminate at what we call the collecting duct, which will then move on down to the renal papilla, and then they'll dump into the minor calyx, into major calyces, into the renal pelvis, and out the ureter. Hooray and a hip hip. So if I were to look at what's going on, first thing that we need to be able to do is we're going to basically clean our backpack. So how do you clean your backpack? You take your backpack, you dump everything out that you possibly can. You're going to look through what you have in the backpack, keep the stuff that looks good, get rid of the stuff that looks bad. You'll put the stuff that you want back into the backpack, but usually before you put it in, you do a quick little double check that, yeah, this is all the stuff I wanted. Before you throw away the stuff that you took out, you do a double check to make sure, yeah, this is the stuff I want to throw away. You might need to do a little bit of shuffling at the end, and then voila, backpack is cleaned. That's the process that we do, or that we go through when we make urine. So what we're going to have are those two sets of arterioles that are feeding into the glomerulus. The afferent arteriole, its job is open up the valve. The efferent arteriole, what it can end up controlling is pressure, or its flow. So imagine this. I have two hoses. If I have a hose that's leading into a whole bunch of grates, and then I have a hose that leads out, if I were to clamp up the leaving hose, the efferent arteriole, and leave the one that comes in, the afferent arteriole, fully open, what you're going to get is a buildup inside of where the grates turn out to be. You're going to get a big old swelling. So if I look at this picture, what you're going to get is if I can clamp the efferent arteriole, if I can clamp that sucker down, inside of the glomerulus, we're going to have a buildup of pressure. What pressure? Blood hydrostatic pressure. And that pressure will be so great, it will overwhelm whatever tissue hydrostatic pressures you have, or definitely whatever you have going on in terms of oncotic pressures. And the result will be we'll have such a great pressure inside of the glomeruli that it's going to shove liquid out. What does it shove out? Anything smaller than a protein. So cells and proteins don't leak out, even though they have been known to, and that's because you have kidney damage, and everyone has kidney damage. If you get sick, you'll get kidney damage. So step one, we build up pressure inside the glomerulus using the two arterioles, and the result is we have such a great pressure that we shove everything out smaller than a protein. Step two, what we need to do is look and see, well, what do we want to keep? So this is going to be within the proximal convoluted tubule, and we're going to use active transport in order to pull back whatever substances we are going to say, yeah, that's an important thing to keep, we should probably hold on to it. How's that going to, or how does this work? Well, we're going to have the filtrate inside of the proximal convoluted tubule. We'll have the walls of it, which turn out to be made out of simple cuboidal epithelium. Those are going to use active transport to pull in whatever we need, and then they're going to be pumped right into the peritubular capillaries. Voila, we claimed the stuff back. It turns out that this only works so well, so if we have too much of something being dumped in, we can't reclaim it all back. But we'll reclaim as much as we possibly can. Hooray and a hip hip. The third part, which is a concentration portion, we don't particularly do this that great, but what we can do here is we can start warping the color of your urine, even though that's more at the collecting duct. And the way you do that is we can control water volume. So inside of this loop, what we can do is take a large amount of volume and start to dwindle it down, and thus increase concentration. If you've ever had a bird drop some guano on you, that white stuff that splats on you, that's its urine, because its loop of Henle, or its nephrotic loop, is so efficient that it can suck the water right on out, and the result is they basically urinate solids. The fourth step inside of the distal convoluted tubule is when you double check. Did I put everything back into my backpack that I wanted? Or did I put stuff in there that I don't want? Or do I need to hunt for things I need to reclaim? You double check your mess to see, like, yeah, it's okay to toss all this stuff. Any last minute adjustments that you need to make, this is the location. So this is where drugs are going to be put in. If you have excess vitamins or anything else, we're going to take all that stuff out. Where does this come from? Well, you take from the peritubular capillaries. We can use active transport to shove stuff into the tubes. The tubes are going to be made out of simple cuboidal epithelium again, and then that can be shoved into the lumen of the distal convoluted tubule, which is basically where urine now is. Or we can pull stuff out or whatever we need to do. That's it. So step one, the glomerulus and the Bowman's capsule, we're going to dump everything out that we possibly can. Proximal convoluted tubule, we'll claim back everything that we want. Distal convoluted tubule, let's double check to make sure that I have things where I want them. The last part, which is the collecting duct, all this gets to do is it can suck back water. So it uses a particular, if given the appropriate signal, what you'll end up doing is forming these things called aquaporins. Aquaporins will bind, and the result is we will have water reclaimed. It's not universal that will happen, but if we have the appropriate signals, we'll start to reclaim water in the collecting duct. From there, we're practically on our way out. So urine's going to flow from collecting duct to the renal papilla, papilla to the minor calyx to major calyx into the renal pelvis into the ureter, flow on down until it reaches the urinary bladder. Your urinary bladder is going to start to stretch. It's going to fill up with urine, it's going to start to stretch out. One of the things that will happen is if it's stretched enough, it will trigger a reflex. So we'll send a sensory neuron sending information to your spinal cord. Spinal cord will have an interneuron to say, hey, wait a second, this is bad. It's going to send the information back out, which will then articulate with the urinary bladder, and that will trigger a contraction, and the result is we'll start to push urine out. What that's going to do is hit the first of your sphincters, which is called the internal urinary sphincter. And what that does, when it hits it, the sphincter will open up, urine will fall to the external urinary sphincter. You're going to feel that, and that's what potty training turns out to be. Potty training is teaching you to control that external sphincter. You're going to sit there and say, oh, I need to pee, but you don't yet. Until you get to about 2/3 of the way filled with the bladder with urine, you can actually control this. It's just once you get past that 2/3, you're probably not going to be able to control it anymore, and it's going to open up and we'll have a mess. But now you know how you make urine.